The textbook defines identification of a microorganism as characterizing a population of cells that descended from a single parent cell into a group of cells. Identifying a microorganism can be done using many different methods. You can use microscopic examination, which is using a microscope to identify different characteristics. You can use the characteristics of a culture that has been grown using the streak plate method. You can also use the results of various biochemical tests done on the microorganisms. Finally, by using nucleic acid analysis, you can identify variances in the microorganism's DNA and RNA. All organisms can be classified into three main groups, and these are the bacteria, the archaea, and the eukaryotes. Some examples of bacteria are E. coli, Streptococcus, and cyanobacteria. Some examples of archaea include halophiles, methanogens, and thermophiles. Some examples of the eukaryotes are animals, plants, and fungi. Additionally, organisms are classified based on a taxonomic hierarchy with the broadest to most specific classifications in order being domain, kingdom, phylum, class, order, family, genus, and then species. Nomenclature is how organisms are named in the scientific field. They are named based on their genus, species, and strain. First is the genus, and it is always capitalized. Second is the species, and it is always lowercase. Then last is the strain, which is commonly a number or combination of number and letters. The name of these organisms is always italicized when typed or underlined when handwritten. For example, Escherichia coli 0157:H7. The name Escherichia is the genus, coli is the species, and 0157H7 is the strain of this bacteria.
Guys, for the CLC project, for my portion, I'm going to be talking about identification based on phenotype. So this is going to entail microscopic morphology, culture, and serological characteristics, as well as metabolic capabilities. So going into it, we're going to start with our microscopic morphology. This is what allows us to be able to identify a microorganism, and this is really important to allow us to determine its shape, its size, as well as other characteristics. So pretty much there are three subtypes that I'm going to be going through that allow us to properly identify and organize these microorganisms into categories. So these allow us to properly identify them as well by using different tools, which I'll talk about in the following notes. So first, we're going to have size and shape. So the way that size and shape is determined to identify these microbes are by using a microscope, and this allows us to be able to identify if it's like a prokaryote, a fungi, or a protozoan, as well as a bacteria. So the microscope just pretty much allows us to be able to properly identify what it is, so that you could kind of tackle it from there from a physician standpoint. Next, we have gram stain. So this allows for identification of a gram-negative or gram-positive bacteria. By using a microscope, it allows the technician to narrow down options in order the proper testing necessary to diagnose a patient. So pretty much from a healthcare standpoint, by being able to just identify if it's gram-positive or gram-negative is really important because we're able to properly treat our patients and kind of figure out what's actually going on just based on being able to identify if it's negative or positive, just because it will entail different treatments. Excuse me, sorry. And then lastly, we have our special stain. So by using a specific stain, it allows for distinctive features of the microorganism to be identified. So pretty much based on the microorganism that you're kind of looking at, the stain can be affected negatively. So it's really important to make sure you're using those stains properly so that you're really able to see what exactly you're looking at just based upon the stain, which is pretty cool that the color can help you to determine that. Okay, next we have culture and serological characteristics. So culture characteristics help to give additional help to identifying these organisms. So I'm going to kind of give you guys a little bit of a breakdown to get a better idea of what exactly this entails. And then specific bacterium can have different colors and shape, which allows them to be identified properly. So just like I said in the previous slide, that a specific shape and color or size can help us to really be able to identify those bacteria and figure out what they are, which is really important when it comes to being able to treat patients properly, because without being able to identify these bacteria, we're not able to really know what's going on, which is really important. Specimens need to be taken from the origin of infection for the microorganism to be identified. And I do have an example for this. So if a patient has a sore throat, we'll go in, swab their throat. And after that swab is taken, it's going to go get sampled, tested in the lab, everything like that. And then if B hemolytic colony was present on the culture, this would allow the physician to know that this patient would have streptococcus pyogenes, which is the bacteria that causes strep throat. So just even being able to take something from that origin, which was the sore throat, and just swab it and be able to help differentiate that, it's really important to actually like go in, take the swab, figure it out from there, which I think is pretty cool personally. But yeah. And then this also allows the physicians to take those physical samples and be able to identify what's going on and the different types of bacteria that are used for the cultures. And then the image on this slide shows the bacterium as well as them cultured on the Petri dish, which I think was super fascinating because it's not something that I had seen before of strep throat especially. And then for the use of serological characteristics, this will allow antibodies to be detected, and it looks for specific proteins and polysaccharides. And then all of these are used to help identify these phenotypes, which I think is very interesting and super fun to learn about. Thanks, guys.
This slide explains the metabolic capabilities of a microorganism. So a colony is a group of cells that come from one single parent cell. The metabolic capabilities of a microorganism includes the end products that are being made as well as the sugars that are being fermented. These identifying factors and traits of microorganisms allow researchers to better understand the importance of the differing traits. Tests can also be done that fit under the metabolic capabilities to help differentiate microbes. One example are biochemical tests, and these tests allow enzymes to be detected and allow metabolic capabilities to be isolated, which is one of the most important methods for microbial identification. And this allows the organisms to be detected at a much faster rate. Identification based on genotype. Genotypes are the genetic makeup of an organism. Bacterial typing is used to further clarify the population dynamicity and track the spread of microorganisms. This helps with detecting nucleotide sequences, sequencing ribosomal RNA, and genome sequencing determining the phenotype. This involves the use of conserved sequences with phylogenetically informative genetic targets. Detecting nucleotide sequences. The key to detection of specific nucleic acid sequences is base pairing between complementary strands of RNA and DNA. At high temperatures, the complementary strands of DNA separate, yielding single-stranded molecules. Nucleic acid hybrids can be formed between two strands of DNA, two strands of RNA, or one strand of DNA and one strand of RNA. The specific sequence can be detected in total cell DNA by hybridization with a radiolabeled DNA probe.
The nucleotide sequence of ribosomal molecules can be used to identify microbes. This is useful because their relatively stable sequences are found in all organisms. Regions of ribosomal RNA that are variable are useful for differentiating unique species of organisms. Constant regions of RNA allow the design of universal primers. Primers will bind to the ribosomal RNA of almost all microorganisms. Different types of prokaryotic ribosomal RNAs include 5S, 16S, and 23S. 16S is the most useful when it comes to taxonomy. 16S and 18S RNA are called small subunits, or SSUs. Once the sequence of an organism's SSU ribosomal RNA has been determined, researchers can compare it to other known sequences. Whole genome sequencing is a process of analyzing an individual's DNA through a sample of blood. Through the advancement of DNA genome sequencing technologies, whole genome sequencing has become the best method for identifying an isolate. The data collected through whole genome sequencing allows researchers to predict antibiotic resistance of a given isolate. Whole genome sequencing has also allowed researchers to study disease outbreaks. In the healthcare setting, the ability to differentiate between strains of a microbe is important for disease outbreak management.
The slides that I'm going to be talking about are characterizing strain differences. The first one is biochemical typing. Characterizing strain variations is crucial for forensic investigators looking into bioterrorism and other biocrimes, as well as for diagnosing specific illnesses. It is not to be used just in investigations of foodborne and healthcare associated illnesses. Biochemical typing is the process of using biochemical tests to determine the identity of different species of bacteria. Strains can also be differentiated using these tests. A biovar, or biotype, is a collection of microorganisms that have the same biochemical signature. These strains also vary from other strains in that they have specific biochemical properties. Beginning in 1961, a global cholera outbreak was brought on by a biochemical variation of Vibrio cholera known as L-Tor. This biovar's distribution can be tracked as it is readily identifiable from other strains. The second way to characterize strain differences would be serological typing. Combining known HLA antibodies with lymphocytes and traits is the process of serological typing. After adding complement, cells containing antigens recognized by the antibodies will lyse and perish. The patient's antigens will be determined when stain is added in the findings, live or dead. Strain-specific differences in proteins and carbohydrates can be employed as identifying markers. The antigenic types of their capsules, lipopolysaccharide molecules, and flagella allow strains of E. coli and similar bacteria to be identified from one another. The designation of E. coli refers to the antigenic type of its lipopolysaccharide and flagella. The servor, or the serotype, is described as a set of strains that have cell surface antigens distinct from other strains.
Phage typing is used to identify and distinguish different strains within a given species when isolated from different origins or geographical locations. Dilutions are spotted onto a bacterial lawn to see if the bacteria are sensitive to the phages, and if they are, they will lysis or explode and show an area of clearance around the pathogen. A specific characteristic pattern is made and enables researchers to identify and study the specific strain that is lysosed, as well as specifically categorize it. Phage typing allows identification for pathogens such as Brucella, Clostridium, Escherichia coli, Staphylococcus, Bacillus cereus, and Mycobacterium tuberculosis, to name a few. Antibiograms are tables showing how susceptible a series of organisms are to different antimicrobials. Using a series of antibiograms can help track a pathogen's resistance to an antibiotic over time. Individual antibiograms do not actually detect data trends because they are usually a summary of a fixed interval of around 1 year. They are tested in a bacterial lawn with a culture from a patient or pathogen needing testing in a petri dish of agar, then using antibiotic discs to see what the pathogen or microbe responds to or doesn't.
Fungi are eukaryotic microorganisms where hyphae are the general cellular unit of some fungal structures. There are multiple different types of fungi and classifications, but for the most part, they all have a chitinous cell wall and a plasma cell membrane. The chitinous cell wall is a stratified structure of chitinous microfilms within a matrix of proteins, lipids, small polysaccharides, pigments, and inorganic salts. These all provide skeletal support and shape to the cell. And chitin is a polymer of N-acetyl D-glucosamine, and chitin is produced through the cytosol, where the chitin is transferred into linked chains by chitin synthetase. The plasma cell membrane contains ergosterol and microtubules made of tubulin and are selectively permeable to regulate the materials that go in and out of the cell. And the plasma cell membrane mainly contains lipids, proteins, and a small amount of carbohydrates. Fungi are able to live in most habitats, however they do prefer or are mainly found on land rather than in the water or in colder, freezing temperatures. There are different types of fungi which prefer different habitats and have different functions based on those environments. but because they do prefer land, they're usually found in soil or on plants, and those that are found on soil or in plants are usually called decomposers. Because of the environment that they're in, they're able to recycle the carbon and other elements that are around them. Overall, though, fungi do prefer high moisture and warm temperatures as their environment allows.
Fungi reproduce either sexually or asexually, and if it is a perfect fungi, then it would reproduce both asexually and sexually, and an imperfect fungi would only reproduce asexually. So the sexual reproduction section, there's two different ways a fungi can go about reproducing. So there's homophilic, which is an independent, and they reproduce on their own, and there's heterophilic, where it's merging cells and combining their insides and creating a new nuclei, and that eventually leads to the formation of new and unique spores. They all follow somewhat a similar process when reproducing and emphasize genetic diversity, and the reproductive flexibility allows fungi to make smart decisions for survival, choosing whether to have offspring independently or with a partner, whether they want to be homophilic or heterophilic, ensuring a varied and resilient next generation that can thrive in different and diverse environments. As for asexual reproduction, they have many different ways of making themselves without a partner present. So yeast, for example, can make babies by growing a bulge on the side of themselves, and then once that bulge gets big enough, it breaks off and becomes its own cell. The most common way that fungi make more is by creating tiny copies called asexual spores. The spores are like mini versions of the parents, and they help spread fungi to new places. So there are many different types of these spores. Some are released from the tips or sides of the fungal threads, like yeast, and some break a piece of the thread, and others bud off from the parent cell. Each of the methods helps fungi grow and learn and explore new environments. Ascomycota fungi have sac-like structures as seen in the picture called asci. They are found in various habitats, including soil, decaying organic matter, and forming symbiotic relationships with plants. Some also inhabit human-made environments. Basidiomycota are fungi with club-shaped structures known as basidia. They are often found in terrestrial ecosystems, particularly in forests and grasslands. They are associated with decaying wood, soil, and mycorrhizal partnerships with plants. Zygomycota fungi create sturdy spores known as zygospores. These fungi are commonly found in soil, dung, and other organic substances. They are widespread as saprophytes, playing a role in breaking down and decomposing of organic matter. Identifying the type of fungi in medical situations is essential in guiding appropriate treatment, predicting outcomes, implementing preventative measures, monitoring resistance patterns, and contributing to broader efforts in public health and medical research. The Bible verse that we chose was from the 1 Corinthians chapter 6, verse 19 through 20. Do you not know that your bodies are temples of the Holy Spirit who is in you, whom you have received from God? You are not your own, you were brought at a price. Therefore honor God with your bodies. So knowing what is in our bodies and how to treat our bodies, and treating our bodies like a temple even when something goes wrong, is just a way to honor God.